Hello, welcome to your lecture on the neurobiology of movement. This lecture will cover three areas related to your textbook reading. First, we're going to answer the question, what is skeletal muscle? In the process, we'll cover anatomy, the physical basis of muscular contraction, sensory feedback from muscles, and so on. The second question we'll address is how the spinal cord controls movement. In so doing, we will cover the monosynaptic stretch reflex and contrast and compare this with the polysynaptic reflexes. In the third part of this lecture, we're going to talk about how exactly the brain controls movement, focusing specifically on cortical structures, the control of movement through descending pathways, planning and initiation of movement, focusing specifically on the association cortex, and also subcortical structures involved in the brain level control of movement. We begin with skeletal muscle, anatomy, the physical basis of muscular contraction, and sensory feedback from muscles. First, it's important to know that the animal kingdom is made up of species that navigate their physical world by swimming, by quadrupedalism, that is walking on four legs, either on the ground, terrestrial quadrupedalism, or in the trees, arboreal quadrupedalism, and in rare instances, bipedalism, as is the case with humans. In any event, vertebrates are afforded skeletal muscles that are responsible for coordinating the physical actions, at the basis of which, or foundation of which, are the actions of specific muscle fibers that make up the skeletal muscles. These include the extrafusal muscle fibers and the intrafusal muscle fibers. Both of these muscle fibers consist of a bundle of myofibrils and overlapping strands of actin and myosin. Whether an animal is moving quadrupedally on the ground or in the trees or bipedally for that matter, the movement of the limb away from the body is called extension, and the movement of the limb toward the body is called flexion. In this illustration, we see a close-up of the anatomy of skeletal muscle. A cutaway of the tissue attaching the muscle of the arm to the bone is shown on the left, with labeling of the Golgi tendon organ inside in brackets. An image of the muscle of the arm attached to a bone is shown in the middle. So you can see how the muscle can operate flexion versus extension, movement of the arm. To its right, you see an enlarged view of the muscle shown, and here you can tell that it's shaped like a flexible or open coin purse. If you look into the coin purse, you can see that the extrafusal muscle fiber is shown on the outside and is served by the axons of the alpha motor neurons. By contrast, the intrafusal muscle fiber is shown on the inside of the purse shape. It is served by gamma motor axons and a sensory axon. It's important to note that the intrafusal muscle fiber is found within the muscle spindle. There are free nerve endings, also the Pacinian capsule, and additional extrafusal muscle fiber. In this illustration of the physical basis of muscular contraction, we can see a cylinder-shaped bundle of extrafusal muscle fibers at the top. A cutaway of its top layer leads to the image of two rows of actin filaments. Each of these is shaped like a rectangular strand with a myosin cross bridge between them. This myosin cross bridge is composed of fin-like structures on the extrafusal muscle fiber. As the actin's filament strand moves to the right, myosin attaches to actin strands, bends in one direction, detaches itself, bends back, and reattaches to the actin at a point farther down the strand. This movement is indicated by the right arrows that you see in the illustration. An arrow on the extrafusal muscle fiber points to the left, for instance. Muscular contraction basically comes down to depolarization of muscle fiber triggers of the muscle contraction, allowing myosin to slide or roll along actin, producing the contraction. The strength of a muscle contraction, as you will note from your reading, is determined by the average rate of firing of various motor units. Sensory feedback is supported by sensory nerve endings that are sensitive to stretch and contained by intrafusal muscle fibers. These sensory endings detect muscle length. Here, the Golgi tendon organs detect the total amount of stretch. I include this illustration here or screenshot to remind you that your textbook publisher has a number of really helpful simulations on muscle structure, the sliding filament, calcium and ATP, also muscle cells and their movement that are worth following up on. Now we will discuss the control of movement by the spinal cord. As you will note from your reading, the classic monosynaptic stretch reflex is the patellar tendon reflex, which is illustrated here. It is an example of movement control via single synapses, where the muscle contracts in response to being quickly stretched, in this case by the mallet hitting the patellar tendon. This is thought to support our maintenance of posture and our ability to adjust our posture and maintain our body position, particularly in a state of tripping and falling. While the monosynaptic reflex includes a single synapse and is what makes up simple reflexes or instinctive locomotor responses, the polysynaptic reflex involves lots of synapses and much more complicated reflexive behavior. It is therefore normally controlled by the brain, and examples include our pain responses and our startle reflexes. In this close-up of the monosynaptic stretch reflex, you can see that only one synapse is encountered along the route from the receptor, where the stimulus is received, to the effector, where the movement is created. Afferent impulses starting at the muscle spindle pass through the dorsal root ganglion and are conducted to terminal buttons in the gray matter of the spinal cord. These terminal buttons synapse on an alpha motor neuron that then innervates the extrafusal muscle fibers of the same muscle after passing through the ventral root. So the entire arc is handled at the level of the spinal cord. In this second illustration of a monosynaptic stretch reflex, you can actually see how the muscle spindle, the red muscle fiber in the illustration, changes in shape based on the flexion or contraction of the forearm. In this third example of a monosynaptic reflex, we see a cross-section of the leg revealing the calf muscle. In the first image, you can see the person is standing upright. So this is the image farthest to the left. The gastrocnemius muscle, the calf muscle, is exposed and labeled. In the second image, the person leans forward. The large calf muscle lengthens. Muscle spindles fire. Alpha motor neurons are stimulated reflexively, and the muscle contracts, causing the calf to bend forward, exerting force at the front of the foot. In the third image, you can see that the stretching brings out compensatory muscular contraction that pushes the toes downward, effectively restoring upright posture. And so by the end of this sequence, the third image resembles the first image. So now we're on to the polysynaptic inhibitory reflex. Immediately, what you should be able to tell here is that besides the usual sensory neuron, we have a variety of connections, including an alpha motor neuron and an inhibitory interneuron, right? So there are more players in this scenario. However, similar to the monosynaptic stretch reflex, afferent impulses starting at the muscle spindle are shown to pass through the dorsal root ganglion and are conducted to terminal buttons in the gray matter of the spinal cord. These terminal buttons synapse on that alpha motor neuron, which innervates the extrafusal muscle fibers on the same muscle after passing through the ventral root of the spinal cord. In addition, you can see Golgi tendon organs with terminal buttons are shown to synapse on spinal cord interneurons. These interneurons synapse on the alpha motor neuron serving the same muscle. The terminal buttons are shown to produce inhibitory postsynaptic potentials on the motor neurons. They are labeled inhibitory interneurons and are shown between the interneurons of the spinal cord. So far, we have considered the control of movement at the level of the spine, and now we're going to consider movement control by cortical and subcortical structures of the brain. Here we see an illustration of the motor cortex and a homunculus, by which you are probably very familiar with now that we have finished discussing sensation. So the primary cortex, which is illustrated in pink, is responsible for causing movements of particular parts of the body. As you know, each movement is caused by contraction of several muscles. There are complex neural circuits located between neurons in the primary motor cortex and neurons in the spinal cord. One important thing to note about the motor cortex is that it shows somatotopic organization, meaning that there are specific parts of the motor cortex that correspond to specific parts of the body. And the image that you see has those components labeled. It shows the primary motor cortex extending from rostral to the central sulcus. and the premotor cortex going from the rostral end to the primary motor cortex. It also shows you the supplementary motor area, which is a region in the dorsomedial frontal lobe, rostral to the primary motor cortex. And you can also see the pre-supplementary motor area. The supplementary motor area and the premotor cortex are important for the control of movement. The SMA is located on the medial surface of the brain. By contrast, the premotor cortex is located mostly on the lateral surface of the brain. Movement is controlled through descending pathways that are both lateral and ventromedial. With respect to the lateral group illustrated here, you can see that the lateral group is primarily involved in the control of independent limb movements, especially of the hands and fingers. This descending pathway includes the corticospinal tract, the corticobulbar tract, and the rubrospinal tract. With regard to the ventromedial group, you can see that it controls automatic movements such as gross motor movements of muscles of the trunk and coordinated movements that are needed for the maintenance of posture, as well as locomotor movements, walking and running. It includes vestibulospinal tracts, the tectospinal tracts, the reticulospinal tracts, and the ventral corticospinal tract. This illustration is worth studying because it lays out the major groups, their tracts, the muscle groups that they affect change through, and the ecological functions or behavioral functions of those muscle groups. So you can see the lateral group and the ventromedial group have different tracts that they involve, and different muscle groups that overlap with specific body parts and have a wide array of postural, gestural, and locomotor functions. Our ability to plan and initiate complex movements is due to the motor association cortex. This includes the supplementary motor area, which is involved in learning and performing behaviors that consist of a sequence of movements, sometimes guided by sensory information. It also includes the premotor cortex, which is involved in learning and executing responses that are signaled by the presence of arbitrary stimuli, as well as imitation. Several subcortical structures are also importantly involved in the control of movement. This illustration shows a lateral view of the brain, wherein you can see the basal ganglia shown as part of the midbrain, also the cerebellum shown in the hindbrain, and the reticular activating system shown in the brainstem. The reticular formation is involved in regulating muscle tone, controlling posture and locomotion. It also exerts control over some specific behaviors that we might talk about later on in this course. The cerebellum controls repetitive movements that necessitate accurate aiming and timing. It's responsible for smoothly guiding movements, stopping movements at the correct time and position, and also integrating sequences of movements. Your textbook has a great simulation showing the premotor cortical and subcortical integration of motor functions. Your textbook also discusses how aimed movement, such as throwing of a ball, by a pitcher or professional baseball player or any professional athlete for that matter is the sort of rapid and controlled and timed behavior that is orchestrated by the cerebellum. Besides the motor cortex, the primary motor cortex, and the supplementary motor area and the pre-supplementary motor area, those cortical structures, the subcortical structures, the cerebellum, and now the basal ganglia are really important for coordinating behavior. In particular, the basal ganglia controls a lot of the inhibition of unwanted movements. And it's therefore the target of interest when it comes to neurodegenerative disorders like Parkinson's disease and Huntington's disease, which involve problems with dopamine neurotransmission in the basal ganglia. So the basal ganglia receive most of their input from all regions of the cerebral cortex and also the substantia nigra. They influence movements under control of the primary motor cortex, and they exert some direct control over the ventromedial system. This image, which is kind of a complicated and messy image, shows the lateral view of the brain with all the cortical areas labeled. The major pathways connecting the basal ganglia and associated structures are indicated by different types of lines with arrows. Excitatory and inhibitory connections are color-coded for your ease of reference. The frontal, parietal, and temporal cortical regions, as you can see here, send axons to the caudate nucleus and the putamen. Then they connect with the globus pallidus, forming a loop. The globus pallidus sends information back to the motor cortex through the ventral anterior and ventral lateral nuclei of the thalamus, that relay grand central station of the brain, completing the loop. What you can see in this illustration also is that there are direct excitatory pathways shown and indirect excitatory pathways shown. The direct excitatory ones go from cortical areas to the basal ganglia, the caudate nucleus, and putamen. The ventral anterior and the ventral lateral nuclei of the thalamus to the supplementary motor area, premotor cortex, and primary motor cortex. And also from the substantia nigra to the basal ganglia, caudate, and putamen. The indirect excitatory pathway is from the subthalamic nucleus to the globus pallidus. Now that we have discussed motor control at the level of the spine and the brain, we're going to consider complex motor behaviors. So the mirror neuron is probably the most interesting thing you read about for this week's unit on the neurobiology of movement. They are, as you can imagine, a very exciting network of neurons that are located in the rostral part of the inferior parietal lobule and the ventral premotor area. This circuit of mirror neurons is also located in the posterior parietal lobe. In any event, they respond when an individual either engages in, observes, or hears a behavior. They help us understand and imitate the actions of others, and therefore are of great interest when it comes to studies of empathy, as well as studies of teaching and learning, particularly in non-human animals. Reaching and grasping are two complex motor behaviors that are controlled in part by the parietal cortex with significant input, obviously, from our visual system. In the case of reaching behavior, the dorsal stream of the visual association cortex contributes spatial information to the parietal lobe's reaching region. And this part of the brain thereby determines the location of a target and supplies that information in a way that can help coordinate movement to the target. In some respects, grasping is the converse of reaching. They go together like hand in glove. The anterior interparietal sulcus is involved in controlling hand and finger movements involved in the grasping of objects. In humans, the inferior parietal lobule and the ventral premotor cortex represent the primary mirror neuron circuit. The parietal reach region also plays a role in reaching, and the anterior interparietal sulcus plays a role in grasping. While the previous illustration of two hands showed the reaching and grasping of an adult and a child, people who are rock climbers will, in the context of their climbing activity, exert both reaching and grasping functions. As you can see from this image, from an fMRI study conducted in 2005, the brain activation of the anterior intraparietal sulcus coincides with the grasping of the hand. As with many aspects of brain and behavioral function, we have a lot to learn from deficits or syndromes involving impaired function, in this case, deficits of skilled movement, the apraxias. In short, apraxias are difficulties in voluntary movement. Their voluntary and non-reflexive nature corresponds to their categorization as disorders of complex motor function or skilled movement. So limb apraxia involves problems with movements of the arms, hands, and fingers, such as the movement you might use to operate a screwdriver or open a latch. It's produced by lesions of the left frontal or parietal lobes. By contrast to limb apraxia, constructional apraxia involves deficits in the ability to perceive and imagine geometric relationships between parts of things. The results are, as you can see illustrated here, trouble drawing pictures or constructing models or structures. This can be caused by lesions of the right hemisphere, especially the right parietal lobe. This concludes your lecture on the neurobiological control of movement. In an upcoming related lecture, we'll talk about disorders of movement more specifically.